In the last section, we discussed the overall anatomical architecture of the brain. Now we're going to a much more detailed level to discuss some of the key properties of neurons and the structures and chemical processes that give them those properties. The reason for covering this material is that later on in the course we'll be identifying the information processes performed by different anatomical structures and seeing how we can combine information processes to perform cognitive tasks. But how can we be confident that the anatomical structures are actually performing the information processes we're going to assign to them? Part of that confidence is based on understanding that the underlying physiology and chemistry can indeed implement those information processes. In other words, we need to know that our higher level descriptions in terms of anatomical structures can indeed be mapped down to physiology and chemistry. Once we've established that, we can confidently use the higher level information models for what the major anatomical structures are doing, and most of the time we won't need to think about how these processes are implemented at the more detailed levels. The human body is mostly made up of cells with different types in different organs, and neurons are brain cells. A cell is a group of structures and chemicals completely surrounded by a membrane or a wall that separates the inside of the cell from the outside. In most cases, the cell wall encloses a compact volume and the cell has a roughly spherical shape. But as you can see, neurons have a much more spiky appearance. And this spiky appearance reflects the fact that there are large numbers of very specific connections between neurons. The wall or membrane of a cell is made up of two layers of molecules of a chemical called a phospholipid. This membrane allows molecules of oxygen and carbon dioxide to pass back and forth freely, but is impermeable to most other types of chemical. But there are some large protein molecules embedded in this membrane that allow other smaller molecules to pass. And in general, one embedded protein only allows or transports one type of molecule. Some of these types of protein act like a channel, which can open or close, and when open, this type passively allows its target molecule to pass either way. Other types of embedded protein actively transport the target molecules in just one direction. For example, suppose this is a view looking down on an area of the membrane from the outside. This is a conceptual drawing of a protein that transports glucose molecules embedded in the membrane. This glucose transporter protein has a pore in its structure, and different locations in the pore make chemical links with the glucose molecule to move it actively through the pore from the outside to the inside. Here's a somewhat more realistic drawing of this glucose transporter protein. On the left is a view from the side, with the outside of the cell at the top and the interior at the bottom. And on the right is a view looking down on the protein from outside the cell wall. The pore for the glucose molecule is marked by an asterisk. This transporter protein is not just a hole through which glucose can pass. It actively moves glucose into the cell, and in the cell the glucose is the major source of fuel for cell processes. All cells, including neurons, have transporters for lots of different molecules embedded in their membranes. Now, as I mentioned, in most cells, the membrane encloses a fairly compact volume. A neuron has a central body, much like any other type of cell, and this central body is often called its soma. But a neuron has a number of extensions that protrude out from the soma, and these extensions are completely enclosed in the same membrane that also encloses the soma. This photograph shows some pyramidal neurons from the cortex. You can see the central soma in white and the fine extensions emerging in many directions out from the soma. And this is a drawing of a pyramidal neuron with the photograph shown on roughly the same scale. The soma is about 20 microns in diameter and is shaped a bit like a pyramid in the case of these pyramidal neurons, which is why they're called pyramidal neurons. Now, 20 microns is a fairly typical size for cells all through the body. But a neuron has a number of extensions called dendrites that can be up to several millimeters long. And as I mentioned earlier, most inputs from other neurons arrive on the dendrites. In the case of pyramidal neurons, there's one dendrite called the apical dendrite that emerges from the tip of the pyramid and branches extensively. And there are lots of basal dendrites emerging from the soma at different points in various directions, generally perpendicular to the direction of the apical dendrite. And these basal dendrites also branch extensively. Then there's one axon extension that emerges from the base of the pyramid. This axon carries the output of the neuron to other neurons, and it can extend several centimeters or more across the brain, and it branches extensively to target lots of other neurons. Now, all these extensions enclose a volume that can be hundreds of times larger than the volume of the soma. So if you define a cell as everything inside the membrane, neurons are actually extremely large, even though the soma size alone is roughly the same as the size of other cells in the body. Now, pyramidal neurons are very common in the cortex, but there are lots of other types. And this shows some different types, some in the cortex, some in other brain structures. The physical shape of the dendrites is obviously different, and the shape of the dendrites reflects to a degree how the neurons connected to other neurons, because most inputs to a neuron from other neurons arrive on the dendrites, and the dendrites extend into regions where axons of specific types of neurons tend to be located. But beyond physical shape, there can also be significant differences in the chemicals and the chemical processes within different types of neurons. So let's have a closer look. The membrane completely surrounds the neuron. And as I mentioned earlier, the dendrites and the axon are bulges in the neuron membrane. The axon tends to emerge from a region that's raised up a bit from the surface of the neuron, and it's called the axon hillock. The inside of the neuron is filled with a fluid called cytoplasm. This fluid flows freely, so it doesn't maintain any shape to the neuron, and chemicals can diffuse fairly freely through this cytoplasm. Running through the cytoplasm are various fairly rigid fibers called microfilaments and microtubules, and only a tiny proportion of these fibers are illustrated in this drawing. Microfilaments are made up of chains of molecules of a protein called actin, and their main role is to act as a kind of skeleton that maintains the shape of the neuron. So growth of microfilaments drives physical expansions to the neuron, like lengthening its axon, for example. Microtubules are made up of a different protein called tubulin, and they have an additional role of transporting needed chemicals to and from the soma and out to distant parts of the neuron. The transported chemicals are dragged along the outside of the microtubules by other chemicals that are called motor proteins. In the soma, there's a nucleus, and it contains genetic information in the form of DNA. The nucleus is itself surrounded by its own membrane, which is a bit different from the external membrane, but has a similar role of managing chemical access to the DNA. Then there's a structure called the endoplasmic reticulum. This is effectively an extension of the nuclear membrane, And in fact, although I haven't illustrated this, it reaches out into the soma, the dendrite, and the axon, and it's important for the synthesis of chemicals in all those locations. One part of the endoplasmic reticulum is called the Golgi apparatus, and this Golgi apparatus is involved in the synthesis of chemicals which the neuron uses to communicate with other neurons, chemicals called neurotransmitters. Ribosomes are structures attached to the endoplasmic reticulum, and both close to the nucleus, as I've illustrated, but also located far out in the dendrites. These ribosomes are where proteins are synthesized using genetic information derived from the nucleus. Also within the neuron, there are lots of free-floating structures of different types, each one completely enclosed in its own membrane. One example is the mitochondria, where the food molecule glucose is consumed to create a chemical called adenosine triphosphate, or ATP. This ATP supplies the energy that drives most of the chemical processes in the neuron. And there are mitochondria in the soma, but there are also mitochondria all through the dendrites and axon. The deeper you look, the more complex the structure of the neuron appears. A huge amount is known about neurons at ever-increasing levels of detail, and I'll be getting into a tiny but carefully selected segment of this knowledge. And as we discussed earlier, understanding can only be achieved by finding the right simplifications and approximations so that understanding is intellectually feasible.